Hi, I want to thank the organizers for inviting me to speak. I am a clinical and biochemical geneticist, and today I'm a genetic counselor, so I get to do both roles. So I'm hoping to explain to you a little bit about what genetic counseling involves, how it can help you as providers, patients, families to understand what's going on. Okay. So I want to just start by saying what genetic counseling is, because I don't know that everyone really knows what it entails. Obviously, people think, well, you talk about the genetics. Well, yes, it's a little bit more in depth than that. So genetic counseling can be done by providers that are either genetic counselors, they have master's degrees in genetic counseling, and they are board certified by the National Society of Genetic Counseling, or by someone like myself, who is a clinical geneticist, an MD or an MD PhD, who's board certified in that by the American Board of Medical Genetics. So it can come at it by either way. And sometimes, by the way, we work together. So I often work with a genetic counselor when I'm meeting with families. Why people get referred to us is really anything and everything. You can have a suspected diagnosis, you can have a known diagnosis, or you can have a family history of a diagnosis. I see the really great way to say it is cradle to grave. In other words, every age, every person. There's no limit to who I see. I follow patients from lifespan. When you come to a visit for genetics, even when sometimes we know what you're coming for, I'll tell you what an appointment with a geneticist or a genetic counselor looks like. We do a pretty detailed medical history. In medicine, we talk about the review of systems, which is how we kind of go through eyes, ears, nose, throat, lungs, hearts. That's actually our entire history. We don't use it as a review of system. We need to know everything because genetics obviously impacts every aspect of your life and every aspect of your health. We also do, there we go, a really detailed family history. And although maybe you're coming to see somebody because you have Wilson's disease or you have a family history, but other conditions can occur. You're allowed to have more than one diagnosis in your lifetime. That's really important to say is that although we're looking at, again, I'm going to use in this example Wilson's disease, what if you have something that doesn't fit with Wilson's disease? So we need to know that because maybe we are looking at something else that's modifying your disease or making it different. So you can see here my example that I've given here is a patient with Wilson's disease. Our squares are males, by the way, circles are females. Just we use this as a very standard diagram, pedigree. But you can see we're looking at things like breast cancer. We look at learning disabilities. We look at other things to make sure that we're not missing something. And then we do pre-test counseling. So if we're going to do testing on you, depending on what we're doing, we're going to do something called pre-test counseling. And what that tells you is when we do a genetic test, what are the possible outcomes? And I'll talk a little bit more about that in a bit. The other thing we talk about, by the way, is discrimination. So genetic discrimination. So my counselors are way better at this than me, but I'm going to give you the gist of it to say that if you have a genetic diagnosis, there are potentials for discrimination in health issues. So we have something called GINA in the US. The GINA is a Genetic Information Protection Act. So that if you have a genetic diagnosis, you are not supposed to be able to be discriminated against for health care. However, loophole, long-term medical care, life insurance, those are not protected. So if you are found to have a diagnosis like Wilson's disease that will impact your later time in care and health, you have the potential to be discriminated against for long-term health care and for life insurance. So we want people to know this because once you get that onto your record, we can't make it go away. However, beforehand, you could go and actually establish these insurances before that diagnosis comes through. So we just want people to be aware of this. And then there's post-test counseling, which is where you come and you sit and we go over the results and tell you what it means and what it all means for you, for yourselves, your families. So I kind of approached this talk today about how I actually talk to patients when they first come and sit down with me in my clinic and how I do genetic discussions. I will tell you, by the way, my high school students are my favorite patients because they're like usually in biology and they're really excited to hear about it, like real world. This is like school coming to life for them. So I just had this experience this week and so it made me super happy. So genes are basically coding instructions for the body. They make everything we are. This is what it's from. We have approximately 20,000 genes in the nucleus of every cell. So there you go, back to your biology. Okay, we're back to high school biology here. And so we have 2 copies of every gene. The exception is the X and the Y. So for males in the room, you only have 1 copy of the genes on your X chromosome. For the females in the room, for the most part, you have 2 copies of everything on your X chromosome. Half of these obviously coming from mom, half coming from dad, the egg or the sperm. Oops, sorry. And genes are coded in a sequence of letters. You and I look at this and you go, I have no idea what this means, but the body can read these like a sentence. I often tell people, if you are familiar at all with coding on computers, zeros and ones, we use A, C, T, and G. The end result is a protein. So this combination of letters makes out various proteins. I often we use a lot of analogies in genetics when we're trying to explain genetics to patients and families, often equate it to a recipe. If there's an error in your instructions, or if you leave out an ingredient, then your product is not going to be right. What comes out of the oven isn't going to be right. Okay. So what are variants? These are also known as the mutations. We tend to use the word mutation less these days, and I'll kind of explain a little bit why. So we use the word variant now when we're describing something that is a change in the standard coding of the sequence. And so in the example here, you can say the standard code at the top should have 2 T's and an A, and instead we got a T and 2 A's. So you've changed that, you've varied that coding. The bottom second one you can see is there's actually a deletion, that TAT is gone. So these are variants, these are changes in the coding. They can be inherited from a parent, or they can be sporadic, meaning occurring in the patient that's in front of you for the first time. Very important to know that difference when we're talking about counseling families for recurrence risk. Okay, so these are the 3 classifications, and this is why we say variant now instead of mutation, because when people hear mutation, I bet as I said that word, everyone went, oh, something's wrong, that's bad. That is not always the case. So for example, pathogenic variants are disease-causing. However, let's see if I have anything else. There's also benign. So you can have changes that do not cause disease. These are still variants, but they're not causing disease. And then finally, we have something called variants of unclear clinical significance. And this just simply means we don't know, okay? Just as means our technology has gotten ahead of our knowledge. We can detect the change in the sequence, but we don't know what it does. The one thing I will say about Wilson's disease that's helpful, and I don't have this in every condition I take care of, we can do biochemical studies. We can look at numbers on a chemistry or in a lab to help us know if it actually is disease-causing, but I don't get that in every condition I take care of. So I dug a hole. This seems weird, but this is actually what I use for all of my patients to explain the various possible outcomes for testing. So if I change that U to an I now have I dig a hole. That is a benign change. It is a variant, but it's benign. It does not cause any change to the sentence. You get the gist. If I change the U to a J, now my word doesn't make sense, and now my sentence doesn't make sense. And so this is pathogenic or disease-causing. And then there's the question mark. This means I really don't know what that question mark is supposed to be. So this is what we call a variant of unclear clinical significance. And again, for the VUSs, I can sometimes do biochemical testing. I can look at urine, I can look at blood. I have a few other conditions. I can look at enzymes in some conditions, but it just is variable. Sometimes, again, family studies can help us understand these changes, but not always. So what about the gene itself, ATP7B? It's the only gene associated with Wilson's disease. There are currently greater than 1,000 variants reported. So I said variants, not pathogenic variants. So there's lots of variants out there. Some are benign, some are VUSs, and some are pathogenic. There's about, I think, over 500 to 600, I think, pathogenic listed in ClinVar, which is a database that we use. The H1069Q is the most common variant worldwide, so about a 10 to 40% population frequency, depending on which population we're looking at. There are no genotype-phenotype correlations. There's been suggestions of some. There are studies that suggest genotype-phenotype, meaning your genetic change gives you this clinical picture. The problem is we're talking about a condition that has 2 variants in every person that's affected. So that combination looks very different in every patient, including siblings who have the exact same 2 variants. And there's also probably some likelihood, some modifier genes, meaning other genes within that 20,000 genes that are modifying the ATP7B. And then penetrance. So penetrance. Does everybody who has 2 pathogenic variants get disease? We used to think yes, 100%. Eventually in your lifespan, by the way, lifespan being 70 years. There will likely be changes to this for various reasons. One would be that, so something we were just talking about was newborn screening. As we do newborn screening for more and more conditions, we start to learn more that there might not be 100% penetrance. There also might be modifier genes. I think registries, actually, I listened to talk earlier, those registries will be very helpful. I participate in a very large registry for a number of rare diseases, and that has changed our outlook on what things look like. So all of these will modify it. This is how I look at the world. This is the gene. And this is kind of, we look at this and we say, where is your variant? Where is your change along this gene here? Can be helpful. Sometimes not. I will say that one of the things is that it's important that we know now, because as we go along and learn more, we might be able to look back and go, oh, actually, this specific variant does have this specific feature. So it is very important that we continue to study this and look at these variants. They definitely have population frequencies of these variants, though. And I'm a picture person, obviously. I loved this map because it really showed me where the variants occur. But one of the things that you can see is the most common variant is really everywhere, meaning at least one copy of it in most of the patients in most every country. But there are certain variants that are much more specific to certain areas in certain countries. Again, as we study those populations more, we might get some genotype-phenotype correlations, but we will have to see. Okay, really quick, I want to make sure, because I want to make sure everyone knows what they're looking at when they look at a report. So when you get your genetic test report, there's going to be a couple things. You see the highlighted here. This is a positive report. I learned early on that when I say positive, it means something different than a patient who hears positive. Patients go, oh, positive, that's great, I don't have it. No, sorry. Positive in my world means that you actually have the change that causes the disease or a change in the variant. So I learned very quickly from a patient who told me that. Then you can see the actual change, the variant in the gene, and then it's homozygous or heterozygous. Homozygous means you have 2 copies of that exact same variant, that exact same change. Heterozygous, sorry, this is not a Wilson's disease report, couldn't find a sample one, but heterozygous means you have 1 copy of the abnormal variant. And in the case of the example on the bottom, they actually have 2 different copies of a variant, and so they're called compound heterozygous, 2 different copies, okay? And then the report should also tell you if the variant is pathogenic, benign, or a variant of unclear clinical significance. So all that should be included on your report. I do want to make sure we talk about inheritance. So Wilson's disease is autosomal recessive. That means you have 2 abnormal copies, one, again, that has come from the egg and one that has come from the sperm. Your recurrence risk to parents who are carriers, meaning they have 1 abnormal copy and 1 normal copy, those 2 people together, those 2 carriers together, with every single pregnancy will have a 25% recurrence risk. I don't care if you have 4 kids, all 4 could be affected. So some people go, oh, well, I've already had 1 who's affected, so my next 3 should be fine. Doesn't work that way. It's each time. It's like flipping the coin. Every time you flip the coin, it's heads or tails, no matter what. So siblings of an affected patient will have a 25% recurrence risk, okay? The other thing that I really did want to talk about too was recurrence risk for an affected patient. So a patient who has an autosomal recessive condition, all of their offspring will be carriers. The risk to their child to be affected will depend upon the carrier status of their partner. So your partner can have carrier testing to determine if they're a carrier, to know if there is a 50% risk for your child to be affected, or is there a 0% risk for your child to be affected, okay? So strongly recommend every partner of every affected patient have carrier testing. I actually found this on the Wilson's disease site, and I really liked it. I thought this was such a great illustration of recurrence risk and who should get tested and what their chances are. So please visit the Wilson Disease Association site, and you can see this beautiful tree. And then the last thing, this is just to leave you guys with, and this will be available to you. These are 2 resources to help people to understand genetics, genetic testing. These are not specific. The first one is not specific to Wilson's disease. It's actually a rare disease organization out of the UK that I love, and they talk about all kinds of different genetics, and it's geared towards families, patients, and caregivers. And the second one is actually the Wilson's disease gene reviews that is really great for providers. So if you have a PCP that you really want them to know about what Wilson's disease is because they care for you, have them review the gene reviews for Wilson's disease. It's a really nice overview. Thank you.